unless you put it under the microscope. And the reason that that's happening is that these crystals happen to be shaped like prisms. And we all know that when you take a prism and you shine light through it, and in this case is coming up from underneath, the crystals are refracting the light, which means they bend the light rays. And we'll see different colors depending on how those crystals grew on the slide. If I see something I like, I can take a picture, but the interesting thing is that it's only a small area you're looking at. Right now, under the microscope, you're looking at something about the size of a head of a pin. So that whole slide that I showed you, that's a whole crystalline array that I would have to search through with different magnifications, and hopefully at some point along the way find something that I like. And if I like it, I take a photo. It's then captured on the camera, which is up here on the top. I can then take that image and print it onto medium. I usually use metal, or I use what's called a museum paper. So you would frame that. The metal ones come ready to hang like that. And I can do them in different sizes. So right here, you're looking at crystals of citric acid that I took that photo, and it's probably magnified there about 10,000 times. It's not adjusted in any way in the color. It's not a composite image. That is what you would have seen if you had been in my studio at that time. So that's how I do the work. What I would like to do now, if I can, is to show you a PowerPoint slideshow that describes how I got into this, my journey through this crystalline world that I work in. And I'll also show you a number of images that I took over the last few years and kind of describe what went into each one. So I will do that at this point, and bear with me here. Okay, now I can't see you all. Let's see here. Can you hear me, Rosie, here?
Yep.
Okay, what am I doing? What should I be hitting here? Let's see here.
So it'll be the desktop one, and then you will go ahead and share your screen.
Sorry, I'm not. Okay, sorry. So when I start this, I'm not seeing y'all, so.
Yep, yeah, it'll look that way in the beginning. And then once you start sharing your screen, you'll be able to see the rest of us.
Okay. All right. But I'm not seeing how I can share the screen.
So it should be that middle share screen button. It's highlighted in green.
I'm not seeing that though, that's the problem. I'm looking for that, but I'm not seeing it. Okay, let me do that first.
There we go.
Okay. There we are. I knew we could do it. So can you all see the, let's see here. What do we have this green box that's I'm seeing here?
Yep, everyone can see. So you'll go ahead and start your presentation there.
It's good? Everybody can see? Okay. I really apologize for the technical lack of knowledge here, but okay. So my photography of crystals has really been an encounter with art and science. My background was as a scientist. I was a research scientist for over 35 years. And what I did was along the way, I got a degree in photography, and I'll show you that kind of path that I took. So combining art and science was something that was very challenging for me, I guess you could say, but it's been quite a journey. The question that I get asked more often than any other is, how did you come up with this idea? And I think that question is a good question. But it shows, I think, a little bit about how we tend to think that ideas and create unique ideas come into being. The other question I'm asked is, how did you stumble upon this idea? That, in my mind, is probably not quite as good of a question, because it implies that as an artist or as a scientist, we come across ideas that are just kind of light bulb moments, I call them, where we just pops into our head. Well, of course, that's not really the case. Uh-oh. I have no idea why it's not working.
Whew.
Okay. That's maybe one of those evenings. But so this idea, I think a lot of people have the misconception that science and art are two distinct things. You have the feeling that most folks think of scientists being very analytical, having very left side brain, and artists as only being kind of right side brain, with the thinking of butterflies and colors and flowers. Well, there was a theory that our brains were very isolated like that, as far, or segregated, that's a better word, and that artistic capabilities came from the right hemisphere, and the more analytical from the left. But over the years, since I got started in science and in art, there's been a lot of study on how brain science works, brain biology, how our thinking happens. And although there are areas within our brain that are certainly utilized for one thing or the other, they communicate very readily. And there's also a lot of plasticity in the brain that allows it to grow and build on certain areas.
Lee, I think what's happening is that your screen may be falling asleep, and your remote is not communicating with the screen once it falls asleep. So if you want to do it manually, you can go ahead and go to the next slide. with just, you can try the keyboard or even just the mouse.
Yeah. There it goes. Okay. Now, Leonardo da Vinci was the classic artist scientist. What he said was study the science of art and the art of science. When we look at scientists, kind of characterize them as a typical scientist would be head in the clouds, prone to strange behaviors and outward appearance, and very creative. When we look at a typical artist, got the head in the clouds, prone to strange behaviors and outward appearance, and creative. So obviously there's some similarities here, and the creative part certainly is one of the most important. But when we look at how did I come up with this idea of photographing crystals, I took my background as a research scientist and I took an observation, and that was that crystals under the microscope could be very beautiful. I knew that many, many years ago, and the next question was, could I make this into fine art? And if you look at this kind of thinking, this is what I was doing because I was in the lab a lot. I was a scientist, and we came up with experiments, and we used what, of course, was called the scientific method. The scientific method, as you recall, is where observation and results in a question. You do some background research. When I did background research about crystals under the microscope and could they be fine art, I came up with nothing. Nobody else seemed to be doing it. So that could mean two things. One is nobody else has ever been successful at it, or it's a crazy idea. Whatever, it seemed like an open avenue for me to pursue my artistic side of my brain. Once we've come up with that, at that point, we ask a hypothesis, and those can be kind of an if-then, or if I can photograph crystals properly, then they could possibly be fine art. We do experiments, and in this case, it's been 15 years since I started my quest on the photography of crystals, and so I guess it's been a 15-year experiment. We collect and analyze data, and I've been doing that along the way. How successful? You have certain metrics. Do I enjoy it? Does other people enjoy it? Is it financially feasible to keep doing this? There's a lot of different ways you can analyze the data. And then you decide, can you accept or reject the hypothesis? So one interesting thing about this quest of crystal photography as art is that it started to branch off into other areas. And I became interested in a lot of different things that were related to crystal photography. And crystals, rainbows, hummingbirds, those are all refraction, where nature's creating color based on the refraction of light through a substance. That got me interested in how the physics of light works, how optics work, how color theory works. Why do I choose a certain color pattern? Why is it successful as a fine art piece and others are not? Or why is it successful with the observer? This got into a lot of ideas about quantum theory, about the biology of vision, about pattern formation and pattern recognition. Why do I pick out certain things under the slide? Remember, I told you it was no bigger than the head of a pin. And this got into thinking about complexity theory. I start off with a solution that is completely random of a dissolved substance. It then starts to form crystals and crystal arrays. And that's called complexity theory. There's also chaos theory, which has to do with fractal formations and why we as a species are attracted to fractals. I got into what is beautiful and what is sublime, and brain biology, and the history of science, the history of art, and the history of photography, creativity in science, creativity in art. And the cool thing was that all these things are connected. Now, it may look like I'm kind of ADD here, but over the years, all of these things have started to play a role in my thinking about how I do my art. So this dichotomy of scientist and artist. Let's take a look at another example. We have Da Vinci, and maybe he was just an outlier. Well, turns out there's many others. One that's kind of interesting that I ran across was this fellow named William Henry Perkin. He was a chemist and an artist. So he had pulled this off of doing the the science and the art together. He was looking for a synthetic form of quinine. And this was during the time when Great Britain had a lot of influence in India, and malaria was a big problem. So quinine sourcing was also a problem. So he was one of the chemists looking for quinine, how to synthesize it. Well, he wasn't successful at that, but one of his experiments came up with this beautiful purple product, powder, that he called mauvine. And what he did, being an artist, he saw that that was kind of interesting. He dyed some fabric. It was the only color like that in the world, basically. It was a breakthrough experiment. It was a mistake, but a breakthrough experiment. Queen Victoria happened to really like it. And a fashion craze started off of this mauvine substance that Perkin discovered, and he made a lot of money. So there's an example of a scientist and an artist using both sides of his brain for success. But the more I studied it, the more I learned that these were instances were prolific throughout history. One of them that I saw was this element called crocoite. It was first discovered in a cave in Siberia. Thing that was interesting about it was that it could be made into a pigment and used as a paint for painters. But of course, anything that's sourced from a cave in Siberia is going to be super expensive. But a chemist stepped in, isolated the compound in there, which is chromium, it's actually an element, and started making pigments using this isolate from this crocoite mineral. And chrome yellow was one of those pigments. And since artists didn't have a good yellow, they were all over it and started using it. This is about we're looking in the early 1800s here. Chrome yellow was a favorite of Vincent van Gogh. So without the scientists and being involved here, it's kind of a questionable whether this would have been available for Van Gogh to paint his sunflower series. Another example is the ultramarine, which came from lapis lazuli, which is found in a mine in Afghanistan. Is the only place in the world. It was super expensive. And all through the medieval times, painters using ultramarine, this beautiful blue color, had limits as to how much they could use because of the cost. So what happened was the French Industrial Society around 1800 had a contest asking somebody to make a blue that was cheaper than ultramarine. and a French chemist won the 6,000 franc prize and made what is called French blue or French ultramarine. And this was pennies compared to the huge cost of the original lapis lazuli sourced material. So without that availability, would Picasso have even had his blue period? Another scientific breakthrough that not only led to individuals creating unique art was the ability to put paint into tubes. Before this, this is in the early to mid 1800s, you had to grind your own pigments. You had to make, if you were a painter, you had to mix your own paints. But now they were available in tubes and could be brought out of doors. And bringing them out of doors, of course, the painting in the plein air like this allowed for the Impressionism and the Impressionist movement to get started, which was basically that you could immediately, it was an immediacy to it, a real-time image. It wouldn't have happened without the chemists and the engineers who built those tubes of paint. So Da Vinci's anatomy, there was studies in perspective many, many years ago. Vermeer was a neighbor of a guy named Antoine van Leeuwenhoek, who was a microscopist and ground lenses. And it's probable that Vermeer used lenses and mirrors in his paintings to get this kind of hyper-realistic and view. Photography, of course, is just a mixture of chemists and optical scientists getting together to create the ability to take a photograph in real time. And it influenced how the Impressionists thought. The pointillism, another art movement that got its start with the Impressionists and photography. Like Cubism has connections to Einstein and his theory of general relativity, space-time. The Surrealists worked with the conscious and the subconscious, which is very Freudian, and that was around the same time in the early 20th century, in the 1900s. So it became obvious to me in this during this time that science and art have gone hand in hand throughout history. In fact, it's hard to find anything, any artistic movement that doesn't have some kind of connection to and dependency on science. So I felt pretty comfortable with taking my scientific background and my artistic desires to photograph crystals and combining them. Now, if we go back a few years, this is where I got my start. I'm the guy on the left there. I was always interested in nature and in science. I wasn't very, well, I didn't have glasses at this time and I couldn't see very well. So everything looked a bit like a Monet painting to me, but what I could see were things up close. And that led to my interest in the world of the micro and down at the very small realm of nature. I started off by building a laboratory in my basement. I bought my own microscope with money I saved mowing lawns, and I was hooked. I knew I wanted to be a scientist, and I was fascinated by all things scientific. I went off to school. I went to Emory University in Atlanta, Georgia. I studied biology, and it was there that I took my first photograph through a microscope. It happened to be a rat flea. Now, a rat flea like this one is the one that carries the bubonic plague. Now, I enjoyed taking a photograph like this. I did the, developed the film and did the printing, the whole thing. But obviously I figured out there wasn't much of a market for photographs of rat fleas. And I went on and I worked over 30 years in the laboratory doing molecular biology, biochemistry, and a myriad of different techniques and methodologies throughout my career. But along the way, I really felt like something was missing and I wanted to do photography. So I got a degree during that time I went to photography school while I was working at the University of Washington, got a degree in biomedical photography. This trained me to do the up close and micro photography that I was really interested in. My first photograph through the microscope of crystals was 40 years ago. These happen to be urea crystals, and the reason they're colored there, of course, is because of that prism-like effect. If you look at crystals just under the microscope without the light shining up from underneath, you don't really see too much. They look like you might expect them. But you would take the same crystals and shine a light through them from underneath, the crystals can create all this beautiful color. So here's basically what this setup behind me has that allows me to do this. There's a light source at the bottom of the microscope. There's a polarizing filter on top of that, and that allows the light to be collimated or ordered as it goes up through the crystal. There's then a light condenser which focuses the light up through the stage, and on the mechanical stage is where the slide is, of course. And I can move, when it's a mechanical stage, it means you can move that around so you can view different parts of the slide. The objectives are where the magnification actually happens. I have 4 objectives on my microscope. They range in power from 4 to 40 times. There's then another polarizing filter, and then there's a way that I can look through the microscope and I can actually send the image to the camera as well, which is on top. I use a Canon 5D, which is a full-frame professional digital camera now. That picture of crystals that I first took 40 years ago was done with film, of course. It's now a live view on the screen, and I can actually take the picture from my laptop or from my monitor without touching the microscope. And this is really important because vibration, of course, is a huge problem. So what goes into photographing crystals? So first I got to research what crystals I want to photograph. In this case, these are acetaminophen crystals. And what I want is a crystalline material that people can understand and a part of their lives. So I call it familiar and fun. I don't know if acetaminophen is fun, but it's certainly familiar. The crystalline material has to be available, meaning I have to be able to buy it or purify it. The chemical properties, it has to be soluble in some sort of solvent so I can dry it on a slide like I showed you. The crystalline properties, it has to be able to refract light, and that's called birefringence. Not all crystals, unfortunately, are birefringent. Some things like table salt, for example, is not birefringent. It forms crystals, but it will not refract light. It has to do with the shape of the crystal. So the experimental stage means basically procuring the material, weighing out a small amount, dissolving it in a solvent, and allowing it to dry on a slide. I then search that slide and photograph what I like. Now, what affects the crystal growth? Now it gets a little trickier because the substance, of course, forms different shapes of crystals. Each crystalline material, like snowflakes, for example, are all 6-sided. So different crystals have different shapes. but they'll also be influenced in what shape they are by what solvent you use to dissolve them. The surface is important. So crystals all need to form from what's called a nucleation point, or some point in space that allows the crystal to start building. It can be another crystal, or it can be just a speck of dust in the solution. The rate of crystal formation is critical. In other words, how fast does that crystal grow? The crystals are growing, if in real time, you can almost see them grow under the microscope, but sometimes they can take anywhere from a few minutes to a few days. But that crystal rate is influenced by the temperature and the humidity, plus a lot of other conditions that are here in the studio. The atmospheric pressure is important. That's how big the droplet will be on the slide as it starts to dry. And timing is super critical. Crystals are dynamic. They're drying, they're growing, they're changing, evolving. There's a certain time when looking through the microscope has the best potential to find something that I can photograph. So I have to be available and ready and find what I need under the slide. What affects the quality of the photograph once I take it? Well, the equipment, of course, the optics these are pretty expensive optics in this microscope, and they're designed to take photos like I want to take. In other words, very flat field. If you were taking pictures of cells, you'd have a different setup here. The camera, of course, is important, just like any photographer. A larger sensor or format allows for higher resolution. The microscope itself has to be kept perfectly in line. Taking a photo through a microscope is very different than taking a photo normally in our macro world. The magnification is critical. The higher the magnification, the less depth of field. In other words, how much will be in focus. If it's a very high magnification, it can be very challenging to get the full crystal in focus. Vibration, like I mentioned, I don't want to touch the camera because I'm magnifying any movement that happens. The crystal quality and stability. Some crystals last only moments after they form, and they continue to dry and change and crack and become unusable. Post-processing or enlargement. Remember, I'm starting off with something that is smaller than a head of a pin, and I want to make a print that may be 6 feet in the longest dimension. So the processing and the enlargement and the quality of the image, the resolution, all has to be maintained throughout that process. And then, of course, there's the operator, me, the composition. I have to find something that works as an artist. Because I'm changing magnification, and I'm also changing the space that I'm looking at on the slide, and the crystal is changing, that composition is a sort of a moving target. So let's look at a few of the crystals that I've taken photos of. This one here on the screen is the one behind me. It's called Symphony. It's a citric acid crystal. It's a single photo. It's not a composite image. It was very rare to get the crystals to grow like that. Remember, they're growing on the slide from all different directions, and they usually can impede one another. So this is a very unusual array of crystals. It actually has every color of the visible spectrum in it. So all those angles of all those crystals, some are refracting so that we see blue, the longer wavelengths, some are rather the shorter wavelengths, and some refracting the red or the short, the long wavelengths. So another citric acid crystal, you can see they're always going to be different. I can never repeat anything. This one's called Innocence Lost. You can see here a good example of how nature creates patterns that we're familiar with on the microscopic scale, as well on the macro scale. Blossom, this photo of crystalline acetaminophen, I saw as more of a flower-like structure. Other people look at that and they see it as a canyon. And it really depends on the viewer. The beholder is very important in my work because there's a certain ambiguity there that is unusual. It seems familiar, but maybe not quite so much. And I also know that what I see is probably not going to be what somebody else sees. Another acetaminophen crystal photograph called Snowfall is a great example of what happens when you happen to have the slide in upside down, which I did at the time when I was looking through this crystal array. It doesn't look anything like it does right side up. And it was only by chance that I happened to stop here and kind of contemplate what it might look like and turn it around and say, wow, that looks like trees and snow, or whatever you may think, but that's what it looked like to me. So it's very important to be, after doing this for many years, it's very important for me to keep my mind open and be able to look through a whole crystal array and see potential, where to focus more carefully. Garden Dreams is the first co-crystallization I did. Co-crystallization means more than one crystal forming simultaneously. It's acetaminophen and salicylic acid. I mixed the two together because I got the idea from a bottle of Excedrin, which has acetaminophen and salicylic acid. It also has caffeine, but I tried these two, and it's one of the few that actually allow for co-crystallization. Usually I need to use a pure substance. The purer the better, because other things will interfere with it. For example, acetaminophen is Tylenol, but I couldn't use a Tylenol tablet. I have to purify the acetaminophen. This photograph, called Caffeine 6 AM, is a photograph of caffeine crystals. It is a great example of how we tend to make certain assumptions when we see a scene in front of us. In this case, the assumption that I made when I looked under the microscope was that there was this tremendous amount of depth. It almost looked to me like I was looking over a landscape or interplanetary space. It was quite stunning, but it was all illusion, and it continues to be illusion because there is no depth here to speak of. It's a little thinner than a human hair. And what we see as depth is actually because the crystals are larger at the bottom of the slide than they are at the top. Flourish is a great example of crystals arrays that look like certain types of art, like paintings, or in this case, almost looks like an engraving. It happens to be Truvia crystals, which comes in a little packet. It's a sweetener. It's made from the stevia plant. This crystal does not have a lot of color because the crystals are very thin and they don't allow for the refraction of light through the crystal array. Here's another one that looks like a painting. It's actually vitamin B3. It's called Sapphire Coast, crystalline niacinamide. The problem with taking these photos of these crystals is they're very, very unstable. They form like this, then within a couple minutes as they continue to dry, their whole crystal shape changes. And what would happen would be a crystalline front would come through the slide area where I'm looking and trying to take a photo and completely change it for me. So they were very unstable. I worked on stabilizing them, and I got this photo. It's called Solace. It's also crystalline niacinamide, but I've used some different substances in the solvent mix that allow the crystals to be much more stable. but it changes the shape, it changes the way they look. One of my favorite substances to work from comes from chocolate, which is both fun and familiar. And it's called crystalline phenylethylamine. Phenylethylamine is a substance produced by the cocoa plant. You can buy it as a crystalline powder. Some people take it as a supplement. It's kind of has a feel-good effect. It's works on our neural pathways and to relax us. It's probably one of the reasons we like chocolate. First light is amazing to me in that it looks like there's a shadow and depth to it. It looks like a landscape. Here's another one of phenylethylamine. This one demonstrates what Da Vinci used, what's called aerial perspective in his paintings to create the sense of depth. Here it's all illusion. Once again, all of these photographs are as is, as I saw them under the microscope. These are very thin crystals. There's not a lot of refraction, not a lot of color, but a great amount of depth because we want to think that. Phenylethylamine can also be treated a different way before you grow the crystals. And by heating and then cooling them, it changes the shape dramatically. And these shapes and thicknesses change the colors. It's called promises. When I added a substance called lactic acid to phenylethylamine, I thought I was going to change the pH. I got the idea because lactic acid is in wine, and I thought, oh, wine and chocolate go good together, so I mixed them together. The result was totally unexpected. And in this case, the lactic acid forms an almost like planetary look to it. And this piece was called Sundance, but it really put me onto an idea that maybe I could add this lactic acid and phenylethylamine and get additional images like this. And sure enough, this photo called Lunaresque is a mixture of those two substances. It is the area across the slide is the phenylethylamine, and of course the lactic acid is a super saturated micro droplet within it. It actually will form crystals within that sphere under the microscope. The dark areas, the black areas, are actually negative space, and that means there's no crystal in that area of the slide. I got the idea to look at a substance called tartaric acid because of an individual who told me that crystals grow out of cork of wine bottles. This one called Majestic is crystalline tartaric acid. It looks like a landscape. Tartaric acid, to give you some idea, it took me probably off and on for 6 months to get the crystals to grow into patterns that were what I would call fine art worthy. A lot of experiments, a lot of different things were tried, and then I would go back to the drawing board and think about what I might do next, or just go to a different substance for a while and let it sit and think about it. But crystalline tartaric acid is one of my favorites. Here's another one called Sweet Surrender. This photograph was part of a series in Phoenix Home and Garden magazine this year on amethyst crystals. Resveratrol is a substance found in wine. It's an antioxidant. It's found in grapes and it ends up in wine. A lot of people take it as a supplement. It's supposed to have a lot of health benefits. It enables me to use wine as one of the solvents. And of course, wine will change the pH. It changes the color, not the color, I'm sorry, the pH. It changes the viscosity of the solution. It changes how the crystals grow. It allows for nucleation points, the little bits of that are insoluble material in wine. And it's a great substance to work with. Resveratrol, these crystals actually were grown in a Chateau Saint-Michel Malbec as the solvent. So it gives a vintage to my crystal photos. This one is grown in a Arizona wine from Arizona. It's called Arizona Stronghold. Whispers is the name. And these crystals lasted probably about 20 seconds under the microscope. I was very fortunate to find them, compose the photo, and take the image before the crystals dried down, cracked, and disappeared. This one called Illumination is another resveratrol crystal grown in wine. It was, though, a different way of using the solvents and applying them at different times. It's called counter solvent precipitation. I learned about that when I was working in the pharmaceutical field. So it was taking that information from my science background, and I was able to get the crystals to grow in a branching structure. A new substance I'm now working with is called CBD, cannabidiol. It comes from the hemp plant. This photo is called Cloud 9. These are, once again, a challenging substance to grow crystals, but I will continue to experiment and try different things and see what nature provides. So that's what I wanted to show you from my gallery, but I want to get back to, I guess, where we were with my original question was, can crystals become fine art? And I would have to say yes. For 15 years now, I've been able to do this kind of work. I've enjoyed it immensely. I didn't know at the time if anybody would even enjoy it or like it or look at it. I didn't know if I could grow more than 1 or 2 crystalline patterns when I started. There's still no one else even doing this, but I enjoy it. Like I say, I enjoy it immensely. So it certainly has been a success from that standpoint. A couple other conclusions I've come to is that science and art are not mutually exclusive. They go hand in hand. And in fact, I think there's a synergy when they can be joined together in a project. And it can be creative and productive. Using this scientific approach served me well throughout my artistic career of basically observation followed by questionings and hypotheses and experiments. Photography of crystals is still an experiment in progress. I will continue to do this as long as I can. I enjoy it, and I enjoyed sharing it with you tonight. What's up next for me? I do have an exhibition called Expressions of the West Valley at the Arts HQ Gallery in Surprise. That runs from the middle of October to November. These are on my website, by the way. November 7th and 8th, I'll be in San Diego, hopefully, for the third rescheduling of the Mission Federal Art Walk in Little Italy. I'm a featured artist there, so I'm looking forward to that. December 4th through the 6th in Tempe, they're having the Festival of the Arts this year, and I will be there again. And starting in January, mid-January, through the end of March, I'll be at the Arizona Fine Art Expo in Scottsdale, Arizona. I hope you can come see me there, because that way I'll have my microscope and the whole setup. You can learn more about my work at photographyofcrystals.com. There's 100+ images on the gallery page. And if you have any questions, you can ask me this evening, or you certainly can email me through my website or at lee@photographyofcrystals.com. And with that, I thank you, and I'm open for questions.
All right, at this time, I'm actually going to stop sharing on YouTube. So if you were joining us on YouTube tonight, thank you so much, and you have a great evening. I'm just going to open it up for discussion on Zoom. So once again, thanks for watching. We had a few viewers on YouTube, so thank you for watching tonight, and you have a good night.